Checkpoint G1. This is when the enzyme cyclin P53 stops the cycle of DNA damage. G2 phase. This is when necessary proteins and enzymes are made for replication. Mitosis. This occurs in 4 parts. Prophase. Everything gets ready to divide. Metaphase. Kinetic fibers push chromosomes to the middle of the cell. Anaphase. Contextual fibers break centromeres and push chromatids to opposite poles of the cell. Telophase. This is reverse of prophase and the membrane reappears.
Interphase. At this point, the DNA is in its chromatin form and the DNA replicates. After interphase comes prophase. The DNA starts to coil itself up into visible chromosomes. The copies of each chromosome, or sister chromatids, are connected together by a centromere. During the end of this stage, the nuclear membrane starts to dissolve and the centromeres move towards the poles, and the spindle fibers start to form. Crossing over occurs during this stage. Crossing over is when a homologous chromosome join up together and sometimes share genetic information during prophase 1. Homologous chromosomes are chromosomes that match from both the mother and the father. Metaphase is when spindle fibers are completely formed and attached to the centromeres of the homologous chromosomes. The chromosomes line up down the middle of the spindle fibers along with their homologous pair. After metaphase is anaphase. At this point, the spindle fibers start to pull on their chromosomes, and each homologous chromosome heads towards a different pole. In telophase, chromosomes are now at the pole, nuclear membranes start to form around the chromosomes, and the chromosomes start to unwind back into chromatin. Cytokinesis occurs and the two cells split. These two cells are still diploid at this point. The next 4 phases occur to each of the cells separately. Now we skip interphase this time around because there is no replication of DNA. During prophase 2, at this point the chromosomes reorganize and the nuclear membranes start to dissolve again. Sister chromatids are again joined by a centromere. Spindles start to form between centrosomes. During metaphase 2, spindles are fully formed again and attached to the centrosomes. The chromosomes line up down the middle of the spindles. Anaphase 2 is when the spindles pull the sister chromatids apart and each goes towards a different pole. Now during telophase 2, nuclear membranes start to form around the chromosomes again. Cytokinesis occurs and 2 diploid cells have now divided into 4 haploid cells. During this crossing over process, the homologous chromosomes exchange portions of their chromatids during meiosis to increase genetic variation.
Cytokinesis. The cytoplasm divides. The cleavage furrow formed edges and the cell separates into 2.
These are the steps of cell division.